How do ribosomes know the proper sequence of an amino acid to form a specific protein? Well, they get that information from messenger RNA. So here the ribosome is using messenger RNA to determine the amino acid sequence. So here's my messenger RNA coming into the ribosome to form this amino acid sequence. The messenger RNA is copied from the DNA in the nucleus. So this is a copy of the DNA in the nucleus. So DNA and RNA are nucleic acids. They're made of chains of nucleotides. Ribonucleic acid. These NA words stand for nucleic acids. Okay. Na nucleic acid, Na nucleic acid. That stands for ribo, as in ribosome, ribo sugar. That's what R stands for. And then D stands for deoxyribonucleic acid. All right, RNA and DNA, they use 4 nucleotides, and as far as I'm concerned, they're the same nucleotides, but one has to be a little bit different to confuse everyone. So RNA uses 4 nucleotides, and we're going to abbreviate them G, C, A, and U. DNA uses the exact same 4 nucleotides, and we abbreviate them G, C, A, and T because there's just a little teeny tiny difference between these two. So when I draw my DNA and RNA, I'm going to draw it like this. So DNA and RNA uses the sequence of nucleotides, each of these is individual nucleotides, to form a sequence, and the sequence is the code for the amino acids. And so if you take 3 of these nucleotides, we'll call that a codon because the 3 nucleotides code for a single amino acid. How does the ribosome know which amino acid to use? Well, the codon tells the ribosome which amino acid to use. And it just happened to be the codon for histidine, okay? So I don't expect you to memorize it. Each amino acid has a unique codon. Actually, a few different codons can signal a single amino acid, but there's only one amino acid associated with any codon. So notice my codon is in green, the other stuff is in light green. All right, so here's my codon right here. And notice there's a transfer RNA that comes and matches up with that codon spontaneously because it has the opposite base pairs. We're going to talk about what that means. And so notice the transfer RNA comes in and matches up with that codon because no other amino acid can fit in that space. And then a reaction occurs to where my amino acid is in the proper place, and then we move to the next codon, which codes for a different amino acid, and the sequence repeats. Okay? So that's how that works. So that's how we go from messenger RNA to chains of amino acids. So DNA and RNA are nucleic acids made of chains of nucleotides, and nucleotides are made of a nitrogen base. This is a different base than acids and bases, kind of. So when they talk about nitrogen bases, I don't want you to think about acids and bases. I want you to think of the nitrogen bases that we're going to talk about. Nitrogen base, a sugar, a ribose sugar from the word ribosome, or a deoxyribose sugar, and a phosphate. Okay, here we are multitasking with phosphates again. In pharmacology, they'll talk about purines later when we talk about cancer and chemotherapy. They'll talk about the antipurines, and they interfere with the ability to use adenine and guanine to make nucleotides. Or we might use antipyrimidines, and that interferes with cytosine, thymine, or its twin, uracil. So these are the purines. They're used for both DNA and RNA. There's the adenine and the guanine. You can just remember A and G. That's fine. The pyrimidines are cytosine and then U and T, which are the same thing other than what? What's different? That methyl group. All right. So cytosines used in DNA and RNA, and uracil and thymine are the matching codes. DNA uses thymine, RNA will use uracil. So these are the nitrogen bases. Okay, well that's very difficult to draw. So that's how I draw the nitrogen bases on my cartoons, as little green squares, the nitrogen bases. And we only use 4, A, G, C, and then U and T, depending on whether it's DNA and RNA. All right. There's my sugar, a ribose sugar. And if we're making DNA, then we'll take an oxygen away, so we can call it deoxyribose. That's where that word comes from, ribose, deoxyribose. I don't make this stuff up. And if we take a nitrogen group, one of those nitrogen bases, and put it on the ribose sugar, then we'll have a nucleoside. And so we took our nitrogen base adenine and put it on the ribose sugar to make the nucleoside adenosine. That's where all these words come from. I have a hard time remembering all this. I just want you to see where the words come from. So this is a nucleoside. If we put the G group on it, guanine on it, then we end up with guanosine. If we put cytosine on our ribose sugar, we end up with a nucleoside cytidine. If we put uracil on it, we have uridine. If we put thymine on it, we have thymidine. And that's different than thiamine, which is a vitamin. And I made it deoxy because thymine is only attached to a deoxyribose sugar. So I've drawn a nucleoside for you, all right? Now, phospholipids are detergents, and Tide, it is a detergent, and Tide's full of phosphates. And so that's where the word Tide comes from, nucleotide. And so there's my Tide. Nucleotides are nucleosides with phosphates on them. This is nucleotide monophosphate, nucleotide diphosphate, nucleotide triphosphate. And we can put the nitrogen base adenine right there and end up with adenosine triphosphate, known as ATP. So nucleotides can form these chains. And so here is my nucleotide, any nitrogen base, my ribose sugar, and my triphosphate group. And it can do this with the diphosphates as well. And we can form chains. Well, this is very difficult for me to draw. So my nucleic acids will look just like that, or just like that. All right, so these are chains of nucleotides that we're going to use to form nucleic acids. So here's my nucleic acid. But instead of having square puzzle pieces where anything can match with anything, we've made different puzzle pieces. All right, notice the cartoon of the nitrogen bases. They look like little puzzle pieces. Why? Because only certain nitrogen bases will fit with its base pair. There we go. Notice that nitrogen bases only match in specific pairs. So G is always matched with C. C is always matched with G. T is always matched with A, or A is always matched with U. U is used on RNA. T is used on DNA. So notice G only matches with C, and A only matches with T, or its twin U. Okay? And this happens spontaneously because of hydrogen bonding. So the nitrogen base guanine will only bond with its base pair cytosine. Okay? If you draw it out, you'll notice that it's hydrogen bonding that occurs. G, the bond between G and C is 3 hydrogen bonds. Adenine will only bind with thymine in the case of DNA. 2 hydrogen bonds, that's important to somebody out there. A only binds with U for RNA. So A only binds with T and U. T if the chain is DNA, U if the chain is RNA. So this is a single-stranded nucleic acid. And that's a double-stranded nucleic acid. So RNAs are single chains. DNAs are double chains. So this is how I draw double-stranded nucleic acid. That's how I draw DNA. All right. Or maybe you'll see that drawn like this. DNA is a double-stranded nucleic acid, and they run in the opposite direction. If you put one chain together in one direction, then its opposite chain will be formed in the opposite direction. Okay? So that's why they call them antisense, or opposite direction. So there's double-stranded DNA. And you can see G is bound with C, T is bound with A, C is bound with G, and notice the little hydrogen bonding that occurs. All right? And notice the phosphate groups, those phosphates being chained with each other. make up, like the sides of the ladder, the base pairs will make the rungs of the ladder. But actually, when this occurs, it forms this helix shape with a major groove. The big groove is the major groove, the little groove is the minor groove. That's not important. What I want you to know is that DNA is made of a double strand of nucleic acid, and maybe you've seen this before. It's a double helix. Well, DNA is going to be regulated by proteins, these long chains of DNA inside of the nucleus. These long chains make up these things called chromosomes, and these long chains of DNA on the chromosomes are wrapped around all sorts of regulatory proteins that wind and unwind the DNA so the DNA can be read. Right? And so here, something is unwinding. My proteins are unwinding the DNA and holding it open. And those unwinding, twisting proteins, they have all sorts of different names. Helicase is a type of protein that winds and unwinds DNA. Topoisomerase, topography means how the DNA is wound around something. Gyrase, to twist, like gyrate is to twist. And anything that ends in ase is a protein. All right, so we'll talk in cancer and chemotherapy about topoisomerase inhibitors and helicase inhibitors. We'll talk about gyrase inhibitors. Okay, so now we're going to read the DNA and do something with it. And so we have these regulatory proteins that unwind the DNA to get it ready for something. And here we're going to use a transcriptase to transcribe the DNA. And so this strand of DNA right here, we've unzipped it, and now we're going to make an RNA from it using a transcriptase. So here's my little RNA right here being formed from the DNA. And now we've formed an RNA, and regulatory proteins can take the RNA and move it out. And in this case, messenger RNA is transcribed, moved out of the nucleus to go to a ribosome to form a protein, right? And then our other regulatory proteins, helicase and topoisomerase and gyrase, then put the DNA back together again. All right? Or maybe we're getting ready to go through mitosis and we want to divide the DNA into two strands, and a different type of transcriptase can replicate each of those strands of DNA. And in this case, we've transcribed each strand of DNA, and one of the strands of DNA can be moved out to the other cell via mitosis.